thank you very much to Michael and the organisers for this invitation to speak at this important workshop looking at the interactions between the EU and Australia. Indeed, some aspects of my talk will reflect those specific interactions with Australia today. But I want to give a perspective on the issues around global research infrastructures, and particularly from the point of view of the International Mouse Phenotyping Consortium that Michael has just alluded to, and the various elements that I think were critical in the success of that consortium, made up that consortium, and also some lessons, I think, from our experience in that consortium, which I'm sure will pervade, I hope it will pervade the discussions on EU-Australia collaborations. As Michael has already mentioned, the IMPC's major role is to build the first comprehensive catalogue of the mammalian genome. And I think from that perspective, it's worth considering the genomic and precision medicine, really the big challenges that face us in human disease and the future of biomedical sciences. And if we look across the landscape of human disease, all the way from Mendelian disorders and rare disease to complex disease, we can see that underneath there are many challenges and issues that need to be addressed, all the way from genetic diagnosis for disease, the understanding of mechanisms of disease, the pathophysiology, ultimately all of which will give us a guide to both treatment and individualized treatment, which is envisaged in precision medicine. But in order to rise to those challenges and to secure our ambitions, and also to reduce risks in terms of the future of genomic and precision medicine, we still need to address an issue that poses considerable difficulties, and that is a comprehensive understanding of gene function. And ongoing discovery research into gene function is still required to interpret the genetic basis for disease, from rare to complex disease. And this, in effect, was the context and challenge that the IMPC met 10 years ago. It's the 10th anniversary of IMPC, because much of the mammalian genome is still dark. It was dark 10 years ago, and much of it is still dark today. That is, that we don't know the function of thousands of genes in the mouse and human genomes. We have, and for many more for which we think we know function, our understanding is relatively superficial. The functional consequences of human genetic variation, which lead to disease, are poorly understood, as are the functional analysis of the various elements of the non-coding genome. This is even darker than the coding genome. What is the genetic variation in the non-coding genome that contributes to disease? Overall, the variants of unknown significance in the human genome, which are produced in their thousands every year, those are increasing rapidly, but they're not necessarily being met by commensurate analysis of function. And of course, the theme of my talk today is that the mouse and mouse genetics is absolutely critical to dissect and understand the importance of gene function and genetic variation. And it's on that basis that the IMPC was established. And from its establishment from 2011 onwards, it really had these four ambitions. Obviously, as I've mentioned, to generate a comprehensive genome-wide knowledge of gene function, applying high throughput mouse molecular genetic approaches, which I'll come to in a moment, and through that to illuminate the dark genome, and importantly, to uncover pleiotropy, the multiple functions of genes. Genes don't just work in one tissue, in one organ, in one pathophysiological state. They impact upon a number of different organ and physiological systems. And it is most important in terms of the understanding of human disease, and to have a comprehensive understanding, that we uncover pleiotropy. Obviously through this, to deliver transformative insights into the landscape of disease, but obviously to provide the models and through which we can undertake mechanistic studies, gain therapeutic insights, and ultimately de-risk the future of genomic and precision medicine. Now, the goal, these are in simplistic terms, the goals over the last 10 years for IMPC. They were to generate a null mouse mutant for every gene in the mouse genome, first of all using embryonic stem cells, but more recently using gene editing through CRISPR, to comprehensively phenotype each mutant using standardized protocols. I'll come back to this in a moment. But comprehensive phenotyping is really the watchword. We wanted to look at all of the physiological systems in the mammalian body and really understand what a mutation in a particular gene would do to all of those systems. And through that, determine a baseline for mammalian gene function. And of course, then ultimately provide insights into the genetic basis for disease, all the way from rare disease to complex disease. To do this, we set up a global consortium. This is the current global consortium. It consists of 21 major mouse genetic centers around the globe. And of course, the reasons for setting up such a large consortium is that I'm sure that you can all appreciate that such an endeavor could not really be carried out by a single or even a couple of centers. It required a global endeavor, bringing all of the expertise and critical mass of centers worldwide to rise to this challenge in mouse genetics and genomic and precision medicine. So let me tell you a bit about how we developed our global research infrastructure and a bit about it in terms of its various components, and then go on to tell you something about its impact and how, through the various components that we've developed in this global research infrastructure, we're able to have the significant impact on genomic and precision medicine that we've had over the last 10 years. So and it's a very appropriate time to do this because we're celebrating the 10th anniversary of the founding of IMPC. But the aim was to maintain and expand a worldwide consortium of institutions with the capacity and expertise to address these ambitions, to provide a centralized data center and portal, and obviously to build on the knowledge and expertise of the consortium, but of course, as I've already alluded to create a critical mass which would really drive improvements in the way that we address these ambitions and aims, all the way from new technology, novel approaches, and so on. And as I've already alluded to form a consortium that would deliver the required scale to look at every gene in the mouse genome, but obviously to provide economies of scale as well, eliminating duplication between institutions and waste. And the other point I should add is that the consortium didn't require, it's not an infrastructure with a major substantive or centralized capital investment. Rather, it's a collection of laboratories. They already have, in a sense, locally their own infrastructure to carry out the aims, but bringing it all together to provide the necessary scale was a key element. We want to look at the funding that went into IMPC over the last 10 years. You can see this over on the right. There's been approximately $500 million invested, half of which the lion's share has come from the NIH, who've been a huge supporter of this consortium, and the other half from national funding in the various institutes around the world locally. So in terms of membership of the consortium, we've always focused on diversity and inclusion. Initially, when we were founded, the primary institutional members, they were the institutes who contribute to large-scale knockout mouse production and high-throughput phenotyping. But gradually, as the consortium evolved, we brought in more members who we called expert institutional members. They didn't necessarily contribute to the large-scale production and phenotyping efforts of the consortium, but they provided mutually beneficial research expertise. They engaged in the scientific activities, they provided advice. They also acted as repositories and centers of outreach for both the resources that were created and the data. Everybody worked to commonly agreed phenotyping and IT standards. This was a huge goal of the consortium, to have common standard operating procedures, providing robustness and reproducibility to all of the science that we carried out, and obviously underpinned by an IT structure that could gather all of the information and release it to the community. It is an open access endeavor, and all of the data and the resources is freely available. And of course, membership of the global research infrastructures, I'll come on to later, included at all of the major repositories, the mouse genetics repositories around the world, providing access to live mice, embryos, and sperm. A minor point, but in fact, to provide some centralized funding for IMPC, there is a joining fee for each member of the global research infrastructure. The IMPC is governed by a consortium agreement. It's led by an international steering committee that has a senior leadership team, which is supported by an executive committee. And each IMPC member has a seat on the steering committee. The steering committee is, if you like, the governing body for the consortium, and it reports annually, both to funders and importantly to the International Scientific Advisory Board, which is called the Panel of Scientific Consultants. But I think importantly, this isn't a top-down or simply leadership, senior leadership-led project. A critical element to the project management of the consortium are the working groups, which were set up across all sorts of facets of the program. And each of those working groups is highly inclusive. It involves anybody who wishes to join. The working groups focus on providing novel approaches, technology developments, but also in guiding the leadership and the steering committee in potential strategic developments that could involve technology or be underpinned by operational issues, but also future thinking about where the consortium should go. And I'll come back to this point later on, but IMPC is very much a shared intellectual endeavor, both top down and bottom up, of collectively delivering strategic and operational approaches. Of course, the IMPC wanted to develop the best practice and be an international beacon for standards in mouse genetics, and in particular from the perspective of the IMPC project of developing and applying standardized protocols for production and phenotyping, ensuring reproducibility, but also ensuring that we endeavor to follow the 3 R's in animal research, in particular providing protocols and approaches to mouse genetics that reduce the number of mice that we use, that refine the operating procedures and handling of animals and the phenotyping and so on and so forth. We ensured that we captured extensive metadata on what we did, not just the operating protocols and the data that comes from the mice, but who analyzed the mice, the environmental conditions in the animal houses, and so on. The working groups undertake constantly a review of protocols, monitoring baseline phenotypes between centers, looking for improvements in the phenotyping protocols, new technologies, new approaches, ensuring improved robustness and also economy as well. And ultimately, of course, the application of automated tools for data production, quality control that I'll come on to in a moment, and a periodic review of the statistical approaches that we use on the data to actually identify the significant phenotypes that we found for each gene in the genome. I mentioned the EInfrastructure. That's obviously critical. We're a dispersed global consortium, each mouse institute producing a huge amount of data, which every day is transmitted or submitted to the IMPC Data Coordination Center, which is in fact at my own institute at MRC Harwell. The Data Coordination Center undertakes data quality control. It coordinates with all of the centers and the working groups to ensure data and metadata standards. And after quality control and validation, the data is deposited in the core data archive where statistical analysis is carried out, and that is at the European Bioinformatics Institute in Cambridge. And as I've already mentioned, all data is open access and freely available for download. So overall, this diagram summarizes the information coming from our mouse clinics, our mouse centers around the world, going to the Data Coordination Center for data validation, QC checks. Some statistical analysis and preliminary data is analyzed at the DCC, but usually all of that data, once validated and quality controlled, is transferred to the core data archive at EMBL EBI for statistical analysis. And every year there are several data releases of data. We're currently on data release 15. So I want to change tack slightly and talk about the relationship of IMPC to other research infrastructures across the world. And IMPC from the very get-go embraced the philosophy of clustering with complementary research infrastructures. And I'll talk about a couple of these in detail, but they include Infra Frontier, which incorporates the European Mouse Mutant Archive, the Mouse Mutant Resource Centers in the USA, including the JAX and UC Davis, which in fact are members of IMPC, the Recon Mouse Genetics Archive in Japan, also a member of IMPC, a very strong interface with the big mouse genome informatics developments at the JAX laboratory in the States, and also with Phenomics Australia. We're talking about relationships with Australia, and I'm going to say a little bit about Phenomics Australia, which is also, of course, a very important member of IMPC. And this clustering of research infrastructures ensures worldwide data integration and visibility of the rich data sets that we're generating in IMPC, as well as being a springboard or catalysis for new projects. I mentioned PathBio, which is an Erasmus project within Europe, a training project for looking at pathophysiological mechanisms through the mouse, and it's emerged from all of the interactions and efforts in IMPC. But let me move to Infra Frontier. Infra Frontier is a major European infrastructure supported by the EU for the generation, phenotyping, archiving, and distribution of mammalian models, principally mouse models. And it provides a variety of scientific resources and expertise, expertise to advance the understanding and treatment of human diseases. It's a consortium of leading research institutions. There are 29 partners across 15 countries. And they undertake model development, archiving, and distribution. Obviously, the centers have phenotyping platforms and so on and so forth. But I think it's important to emphasize that if you like, and Martin Rabe, who's one of the directors of EMMA, has often said that Infra Frontier is, if you like, the landing pad for IMPC. It provides across Europe much of the infrastructure that, of course, is being used by IMPC in terms of its specific ambitions to create a complete catalogue of mammalian gene function. And of course, the goals of Infra Frontier are very much aligned with IMPC, all the way from providing knowledge about gene function that's going to impact on a whole variety of biomedical science areas, including COVID-19 research, cancer research, the understanding of the genetics of rare disease, and so on. And in terms of COVID-19 research, it's really emerged out of IMPC, a global consortium for mouse models for COVID-19. Many of the partners across IMPC, through the generation of null mutants and phenotyping, generated a number of models that bear on our understanding of COVID-19, and obviously the use of the mouse as a model to study various mechanistic aspects of host susceptibility and infection and so on. Just another way that a particular infrastructure is acting as a springboard for interaction with other infrastructures and the creation of new infrastructures, collaboration, and consortia. All of the European IMPC partners are members of Infra Frontier, and Infra Frontier partners contribute to all of IMPC activities. And in fact, Infra Frontier and EMMA is the largest distributor of IMPC lines worldwide. I want to briefly move on to and just say a few words about Phenomics Australia, since we're talking about EU-Australia interactions. Phenomics Australia is a very important partner to IMPC. It's obviously part of NCRIS in Australia. And of course, both Phenomics Australia and IMPC have a shared vision through the discovery of gene function and the global mission of IMPC to look for better health, improvements in genomic and precision medicine. And again, as you can see underneath, IMPC aligns with many of the activities of Phenomics Australia, from biobanking to in vivo genomic engineering, creating new disease models, genotyping and phenotyping platforms, including, of course, looking at the underlying pathology of the mice that have been created. And the shared benefits are transparent as well in terms of IMPC fostering coordinated international collaboration that addresses global challenges. There are a variety of points here, some of which I've already alluded to, from aligned strategic goals to development of technologies, shared platforms and tools, importantly, harmonized protocols, increasing research infrastructure visibility. We are a global community of practice that is really working together day in, day out through IMPC, the working groups, the other infrastructures that we cluster to, that provide, if you like, a global network of access to mouse genetics that I think is unparalleled. If we were to go back 10 years, you just wouldn't have seen this. Let me say a little bit about the socioeconomic impact of IMPC. I think this is very important. Before I go on to tell you a bit about the achievements of IMPC, of course, that global consortium reduces repetitive, non-productive and duplicative research, as much as that impacts upon reducing the number of animals used and the 3 Rs. I've already alluded to the fact that one of the key goals is to deliver reproducible and robust data through harmonized protocols. Importantly, as I'll come on to say a little bit about, we employ in our analysis of gene function, we employ both female and male cohorts of mice. This is incredibly important. Genomic and precision medicine has to take account of gender. And one thing that IMPC has done is to illuminate the tremendous sexual dimorphism in gene function that has to be taken into account as we develop new therapies and as we develop precision treatments for disease in the future. The treatments that are administered to males are in many cases going to be different from those that are administered to females. And ultimately, the socioeconomic impact is brought about through extensive outreach to diverse community through our comms working group in IMPC, and of course through the portal and social media. So let me move on to IMPC and just give you a feeling of the impact that we've made. Very briefly, I can't really go into a lot of detail about the scientific impacts and outcome. This just shows you the IMPC phenotyping pipeline. It's very complicated. I'm not going to go in it, but you can see that we have a very large number of tests that bear upon a whole variety of disease areas, from embryonic development to neurology, to cardiovascular, to musculoskeletal, to the immune system. It's that richness of data that we're acquiring on each particular mutant that we're creating that is pivotal to the success of IMPC. And this is the overarching program update for IMPC. As of a month or so ago at data release 15, we were approaching 10,000 genotype confirmed null mutations produced by IMPC. That is, in fact, well over half of the orthologous genome, the genes that are orthologous between mouse and human, and represents a major achievement. We were on target to do this for this year. We've achieved that target. And of those nearly 10,000 genes, most of them have been phenotyped. And the current data release covers over 100 million data points and over 600,000 images, including X-rays, LAC-Z, expression patterns, and so on and so forth. I exhort you to go to our website, mousephenotype.org, for our full publication list. You can see over the last 10 years the novel features of the mammalian genome landscape that have been released through our studies, an extensive new collection of disease models, new candidate genes, new functional knowledge, novel insights into gene function in particular disease areas such as deafness. Pervasive sexual dimorphism has been revealed, which I've already alluded to, and insights into human disease from the analysis of mouse lethal genes. But the most compelling picture that I can provide to you is that wherever we look, novelty abounds. The genome is still really dark to us. Wherever we look, we find novel information about genes and gene function. Because of the wide variety of procedures that we undertake, which bears on a whole variety of disease systems, and we find that many genes have multiple functions. We see multiple phenotypes, and there are very few genes for which we find no phenotype at all. Pleiotropy, the multifunctional aspects of genes, is pervasive. It provides comprehensive insights into the comorbidities, the multimorbidities that are often associated with disease. It's providing a new window into individual human diseases and the pathophysiology of those diseases. I'd like to go back to an analysis that was carried out some years ago. It was reported in Nature Genetics in 2017. It was an early analysis on the first 3,000 or so genes that IMPC had analyzed, and we found considerable new functional knowledge. What was extraordinary, and this is still borne out as we move to 10,000 genes, is that 90% of the relationships between gene and phenotype, between the gene and the potential disease phenotypes, had not been reported before. There's a really dark genome out there. And of the 3,000 or so genes that we analyzed at that point, over half of them had not had a mouse mutant produced, and there was new functional knowledge for over 1,000 genes. That can, as we increase the number of genes we look at, those figures still bear out. Novelty continues to abound as if we look at the mutations that are embryonic lethal or subviable. These are a big class of genes that we look at. And about a third of each of those classes of lethal genes and subviable genes had no previously described phenotype and are entirely novel. If we look at all of our mutants produced from embryonic stem cells, this was recently reported in Nature Genetics, over half of the null alleles that we produce there, no equivalent mutant mouse line exists. And without those mutant mouse lines, we really can't gather and grasp all of the detail about the ensuing pathology, the disease phenotypes, and the pleiotropy. That worldwide impact and outreach, of course, is strengthened by the fact that IMPC has numerous intimate relationships and collaborations with a whole variety of disease foundations and other consortiums across the world, including, for example, the European Joint Programme on Rare Diseases. Much of the IMPC work is focused on delivering information about specific genes to these consortia worldwide, as well as interacting with some of the major biomedical sciences databases. And that impact is reflected in the fact that this year, after 10 years of work in IMPC, we have reached 4,000 publications that have been produced worldwide from people who are using either IMPC data or IMPC data resources. What are the future perspectives for IMPC? Well, if you go to our website, you can see our strategic plan for 2021 to 2030. But as we move from completing the null mutant resource to looking ahead to the other challenges that are going to bear on genomic and precision medicine, we're going to increase our focus on the functional analysis of human genetic variation. We're now well on the way to completing having a baseline of gene function across the genome. Now we want to look in more depth at the individual function of how genetic variants in particular genes or in non-coding sequences bear upon disease. We've already begun to set up a CRISPR pipeline to generate mouse models of human coding variants. This is already going on at many of the institutes across the world. We're beginning a pilot to CRISPR and alter the non-coding component of the genome to better understand how changes in non-coding sequences, regulatory sequences, long non-coding RNAs bear upon disease, all the way from rare disease to complex disease. And importantly, we're exploring more the genetic context and the environmental impacts upon how human genetic variation and genetic variation generally in mammals moves from variants in the genome to disease phenotypes. So I just want to make a few concluding remarks as I wind up. I hope that I've been able to persuade you that IMPC is really transforming through this global consortium the landscape for genomic and precision medicine. It's delivered mutants in over half the orthologous genome and expects to complete the orthologous genome over the next 2 to 3 years. Phenotype data from nearly 9,000 mouse lines. In 2018, we delivered our target of mutants and phenotypes for 1/3 of the coding genome. And this year, we met our next target of half the orthologous genome has been analyzed. IMPC is truly illuminating the dark genome, and through that, it's providing fundamental knowledge that will inform us about the genes involved in rare and common diseases, and it is a key underlying resource for the future of genomic and precision medicine. Last but not least, some thoughts on the lessons learned in terms from both myself and my colleagues of 10 years of this exciting and sometimes very challenging endeavor in terms of infrastructures and what we might reflect on for the future as well. The first thing I think is that infrastructures are people. What drive infrastructures and what's driven IMPC are the people that are involved. It's a community of shared intellectual endeavor. And if you ever lose that in an infrastructure, I think you're lost. And from that perspective, it thrives in a collegial environment that is both bottom up and top down. A mutual respect from all communities within an infrastructure, all of the working groups, all of the leadership that come together to really provide more than the sum of the parts. And momentum is, of course, dependent on that shared collegiate appetite for the infrastructure and the endeavor, which is at the heart of what IMPC is doing. Of course, outreach to funders, stakeholders, patients, and the wider scientific communities is absolutely key, and something that we've put increasing emphasis on in terms of IMPC as the program has gone on. And last but not least, this, I think, key point about infrastructures. Infrastructures often form relationships and clusters to other infrastructures. They don't stand in isolation. They are part of the complex network of scientific endeavor that's going on in a particular area or domain of biomedical sciences or any other type of sciences, or for that matter, the humanities. It's those networks that really provide a springboard for new endeavors, new projects, programs, and infrastructures to emerge. And with that, I'd just like to thank and finish by thanking all of the members and institutions involved with IMPC, and to thank also Michael Dobby and Phenomics Australia, who've very much been a key part of IMPC and a key part of my thinking. I can't possibly put up a slide with all of the names and individuals involved. There are literally hundreds of them, but each and every one has contributed in their own way in what has been an exciting and collegial time for all of us. I think we've all appreciated that as scientists. I hesitate to call it an infrastructure. It's been great science and it's been great fun all the way. With that, I'll finish and very happy to take questions. Thank you.